So welcome, everybody. My name is Michelle Prepisny. I'm the Chief Advancement Officer for the Children's Tumor Foundation, and I'm absolutely delighted to welcome you to our, this time it's our NF2 Schwannomatosis meetup, where we're going to be talking about some of the research advances that were presented at the most recent NF2 state-of-the-art and NF2 and Schwannomatosis state-of-the-art held at UCLA in collaboration with CTF. And that was just this past January 12th through 15th in lovely LA. So delighted to welcome you. It's going to be a really amazing evening. I just have a few housekeeping before I introduce a few people. So first, there is closed captioning. If you need closed captioning, you can click on the closed caption button. I think it's about the middle right of your screen. So we have that for everybody. Please introduce yourselves in the chat. We'd like to know where you're from and what your connection to NF and CTF is. Also, make sure you're muted unless you're speaking. There will be time for questions and answers, and you can put the Q&A, your questions that you have, in the chat, and we'll make sure that we get to all of them before the end of the evening. I want to introduce or just welcome a few patient representatives that we have on the call. All of you are patient advocates and patient representatives, but there are a few people who actually attended, a few patients and family members who attended the NF2 and Schwannomatosis State of the Art, who are going to give their input a little bit later on in terms of the program and the agenda and the research that was presented. So I want to welcome Clayton Molina, Sheila Heel, Barbara Franklin, Camille Toms, and Michelle Holbrook, who is our 2023 National Ambassador for the Children's Tumor Foundation, who's just getting off a plane and will be joining us momentarily. So without further ado, I'm going to welcome and introduce Annette Bakker, who is the president of the Children's Tumor Foundation, who is going to welcome our panelists, Marco Giovannini and Leon Info, this evening, who are the co-organizers of the NF2 and Schwannomatosis State of the Art. So very pleased to have all of them here with us tonight. It's going to be a great presentation. So Annette, I'll hand it over to you.
Thank you, Michelle. Very excited to be here. If I would be a reporter, I would say welcome from Los Angeles. We're going to give you an update on the state-of-the-art NF2 Schwannomatosis meeting. I think the special thing that we have for tonight is that we actually have the co-chairs present. So Dr. Ninfu and Dr. Giovannini from UCLA have been coordinating this exciting NF2 schwannomatosis meeting. So before getting into the questions, and we have been prioritizing some of the topics, and of course, for those patients and other members that have been at the workshop, if there is anything you think we should discuss, please throw it out there, raise your hand and make sure we cover the topic of your preference. But without further ado, let me first go to Dr. Ninfu. Dr. Ninfu, can you just introduce yourself so that people actually know, for those who don't know yet, who you are?
Hi, I'm Leon Info. I am one of the neuro-oncologists at UCLA. I'm also the clinical director to our NF and Schwannomatosis Comprehensive Clinic here at UCLA.
Thank you. Dr. Giovannini?
I'm Marco Giovannini. Hello, everybody. Hi, Annette and Michelle. I am also at UCLA. I'm the scientific director of the UCLA Comprehensive Neurofibromatosis and Schwannomatosis program, which I co-founded with Dr. Nienfu in 2016, and I've been in the NF field since 1994.
I guess you started it too, right, Marco? So we had a little prep call before tonight, and the way we wanted to structure it is have Dr. Ninfu really cover the more surgery versus treatment aspect of the meeting. I think there was a lot of interesting discussion about surgery, when to do surgery, whether surgery was interfering, or let's say whether treatments were interfering with the surgery, and what kind of treatments are on the horizon. So Dr. Ninfu, take it away, your perspective on what you think you want to share with the world about what we learned in LA last week.
Well, I think to start, in the end, the treatment for NF2 related and the other non-NF2 related schwannomatosis, the main treatment is still surgical treatment. And I think we had a lot of discussion among surgeons as well as clinicians like myself, whether patients should be treated early. I mean, we do know that tumors are easier to remove no matter where they are, whether it's a spine, in the cervical spine ependymomas, for instance, with Dr. Michelle Calamaridis from France, who gave a talk, or the vestibular schwannoma scenes with the NF2 related NF2 patients. And Dr. Brad Welling from Boston gave a talk on this. And they both, I think from a surgical perspective, do feel that the smaller the tumors, the better the outcomes. And of course, it is easier for them to remove. However, I think on a medical sense, sometimes we don't know which tumors should be removed. You know, as clinicians, we ask them a lot about how can you predict which ones are growing? How do you know it grows? And sometimes these tumors grow a little bit and then they stop growing. And they are not causing any symptoms. They're not causing hearing deficit or any other neurological deficits. How do we know when we should have these tumors removed and have those patients be referred to a neurosurgeon? And I think kind of the takeaway we got from that is we don't know the natural history of tumors related schwannomatosis very well, right? We don't get a sense of, you know, majority of the time, how fast do any of these things grow? And also, we are reluctant to have patients get surgery when they have no symptoms because there's always a risk of causing symptoms. But at the same time, if we know we have a very good surgeon, and this is usually when it comes down to surgeons that are very experienced with patients with NF2 related schwannomatosis or other schwannomatosis. If we refer patients to those surgeons, their outcome are probably better than other surgeons who try and remove just sporadic tumors and really don't think of these patients in a kind of as a whole, as a patient with a syndrome. You know, that we're not removing just a tumor and then have to not worry about the rest of the tumors later. Versus surgeons who understand patients with schwannomatosis well, I think they understand that implication and would carefully follow these patients. I don't think any of them would choose to remove a tumor right away on their first meeting with a patient, unless we as the clinician say, this tumor is growing very fast and we need it removed now. So, and then I think it requires kind of a multidisciplinary discussion. And even while we were on stage, you know, the nice thing is we had a lot of case related discussions that we had between all of us that included all the surgeons and the clinicians. And I think in the end, all of these patients need that kind of collaborations between the medical doctors as well as the surgeons to really make a decision when a tumor should be removed.
Yeah, and I just wanted to, from a audience perspective, I just want to applaud the NF community. I think it is a very ego-free community. And I'm very proud to be part of that, because there were a lot of cases that were discussed where there was a certain level of disagreement between the clinicians, but it was really a, what would you do in this case? Is this a good idea? Would you do different? And I think these case studies were absolutely fantastic, I think. So, Dr. Nfu, I just want to continue a little bit on treatment modalities, right? So we showed, or let's say the clinicians showed what the clinical trials were that today are in development. Intuit. So a lot of excitement around Intuit. I can tell you, not only sitting in the meeting and seeing the number of drugs today are being treated. We have now a second drug in Intuit called neratinib. What do you see? Where do you see Intuit go? There was a presentation around the vaccines, the VEGF vaccine. Can you comment a little bit on that? what kind of options do you see going forward with Intuit?
I think the excitement about Intuit is that it allowed us to test multiple treatments. I think it was just very exciting that we kind of hit a good drug on our first run, right? So our first drug onto the trial was brigatinib, and there were data presented that brigatinib did lead to tumor shrinkage in several different tumor types in patients with NF2 related schwannomatosis. And I think that in itself kind of created an excitement around Intuit. But I think as a clinician, the bigger thing for me is that it also means I have a platform in order to try different treatments. So right now, the next treatment that we will have on the trial, since the brigatinib trial arm has been closed, so the next arm coming up is neratinib. That's a kind of a pan EGFR HER2 inhibitor that has been shown in clinical work, especially with Dr. Giovannini's lab and other labs, for instance, that can stop and maybe shrink tumor growth in patients with NF2 related schwannomatosis. So that is one option, but a trial like this only really works well if we have a lot of different options to test a patient. It is meant to design as what's called a platform trial. That's where we can enroll patients with multiple different type of tumors, but it's also designed to have an adaptive design, and that we can test multiple drugs at the same time and only move forward things that we think is working early. So the more we have on the study, the more options we have on the study, the better. So right now, like we said, brigatinib is already done. Neratinib is the only other option we have. So one of the physicians from Japan actually presented his data on developing a VEGFR vaccine. So as you know, we use bevacizumab a lot in patients with NF2. That is a VEGF inhibitor, the inhibitor of the actual factor, the vascular endothelial growth factor. And we know with Bev, we have very good results with some tumor shrinkage, with improving in hearing. But at the same time, Bev can come with a lot of side effects also to patients, especially young patients who go on Bev long term. So the idea of the vaccine really is they're trying to create a way for the immune system to trigger itself and go after any tumor cells that have an increased amount of the receptor. That is a protein that accepts the activation from VEGF itself. So it's a receptor, the VEGF receptor in itself. So it's really the immune system, somebody's own immune system that will develop cells that will attack these tumor cells or increase tumor cell growth. And it's exciting because so far it sounds like he has had a small pilot study. And in that small pilot study, he has seen some tumor response in his patients. The difficulty with that is that it can take a while before the patients will have some response, because you can imagine it's not the drug that is going to attack the tumor cells directly, right? It's the immune system. So the drug gets in, and then it has to rev up the immune system to go after any increase in the VEGF receptor. But because of that, they also noticed very, very little side effects from patients who were on this trial. Did not expect any of the side effects that we otherwise normally would see with bevacizumab. So I think that's very exciting to see that type of data. I mean, what we want is a drug that can patients tolerate well, but also work well, right? So, it sounds like it may be a combination of both. And, that would be a drug that I would love to see in a trial like Intuit.
Perfect. Thank you so much, Dr. Info. So here we go. Tumor heterogeneity. I think it was one of the things that definitely came up very much during the meeting. and not look at a tumor as some kind of, football of one cell type, but in fact, looking at a tumor as a composed of different cell types, and where every cell type plays a very different role in the tumor biology. Dr. Giovannini, your take on tumor heterogeneity and what that may do when we think about treatment modalities, when we think about understanding the biology of both, let's say, what we called before the NF2 schwannomas and the painful schwannomas from, let's say, the previously called schwannomatosis. So could you comment on the biology and what was discussed at the meeting?
Sure, that's a great question, Annette, because it's becoming more and more obvious that in spite of looking histologically uniform population of Schwann or meningeal cells, respective for schwannoma and meningioma, which are the main tumor types associated with schwannomatosis in general, not just NF2 schwannomatosis, it is clear that there is a great deal of heterogeneity. So what is heterogeneity? Heterogeneity means that cells, individual cells that compose a single tumor in a patient are not the same. So they depend on different signaling pathways, which means different mechanisms of proliferation. So we now know that there is important heterogeneity within a single tumor in a patient, but also among different tumors in a single patient. So it's easy to understand that the therapeutic intervention that we do by providing a single drug, a single inhibitor, for example, will have different efficacy in different portions of the tumor and on different tumors in an individual patient. So that was obvious already by looking at the result of previous trials for NF2. Unfortunately, we don't have had specific trials for the old name schwannomatosis, schwannomas, but hopefully there will be trial soon, and that's something we might want to discuss, Annette, because it was an important topic at the conference. We observed that the response of a single agent was variable. At most, about half of the patient responded, or at least half of the tumor responded to a single drug. And there was a great deal of differences in response of tumors, of different tumors in a single individual to that drug. So that's the consequence of tumor heterogeneity, which again is different areas of a single tumor rely on different cellular mechanism of proliferation, which then can respond differently to a single therapeutic, to a single drug. So I hope this is clear enough, but that's the idea of tumor heterogeneity. Of course, what we know now, and these are studies in my lab in collaboration with the lab of Dr. McClatchy in Boston, we know that heterogeneity can also evolve. So it's not a fixed situation, but it can evolve during time, during the development of a single tumors. And also it evolves and in response to therapeutics. And that's another point that was discussed in depth at the conference. What is the effect of exposing a tumor, a vestibular schwannoma, for example, to multiple drug regimens in time? Because we know that the tumor that we treat after 3 different drugs, for example, 3 different trials, will be different from the tumor that we treated initially, that was naive, that didn't see any drug before, because tumor cells evolve, they adapt to the situation in which they grow, and they try to escape, unfortunately, the inhibition that a drug can provide, at least in a part of the tumor, as I explained before. So I think these are important considerations, especially in view of the fact that one consequence of drug exposure can also result in it also has implication on the surgical outcome once the tumor has to be removed, because the tumor could be more challenging, could be more vascularized than it was at the beginning of its history. So I think these are important concepts. And again, this view is very new. But we think that it's also highly relevant for NF2 tumors, as it has been showed in most tumors and cancers in other conditions or other sporadic tumors.
So I think you mentioned an important point, Dr. Vanini, is we are still understanding how these tumors evolve. So there was one of the topics also to pick up, let's say, the natural history study that was started years ago at the NIH. Now there is somebody new who kind of says, okay, and I blank on his name now, but somebody who says, okay, we need to reactivate the NF2 longitudinal natural history study in order to understand better what is happening to the tumors. What is, and maybe either you or Dr. Nympho, what is the action that we have all these NF2 accelerators on the call today? I know we are having meetings with the NIH and our people from the NF2 registry to make sure that people are informed that this study is recruiting patients to understand, to be able to treat patients better. Action items.
Yeah, I mean, I can briefly comment, and then Dr. Nympho, I'm sure she has specific ideas on that. But the doctor you mentioned was Dr. Chitiboina Prashant from NIH.
Yeah, I didn't want to say his name because I wasn't sure how to say it.
Yeah, I'm not sure. I just try. But we were lucky to have him at the conference because he basically inherited a project that has been going on for a few years now at NIH. And the idea, the basic objective of the project is to define what happens to specific NF2 tumors over time in the absence or in the presence of a therapeutic intervention. So this is a concept that can be difficult, I guess, to understand, because it basically means let's see what happens over time to individual tumors in a patient, but it is extremely important. And the point I want to make here is that, first of all, it is important to corroborate the initial observation of the natural history study from NIH that were published years ago. And there were issues in that report, and that was discussed at the conference, in the sense that some of the imaging, MRI imaging that was used in that study was not acquired in a way which is ideal for 3-dimension reconstruction of the volume, which is what we absolutely need to monitor the growth of these slow-growing schwannomas and meningioma. So it was a technical limitation, and that led to the description of a specific growth pattern of schwannoma, suggesting that the schwannoma would grow for some time, eventually stop, and then grow again. So that particular growth behavior has been debated over time, and we think it could be explained by this technical limitation that the first study had, because the sense in the audience was that tumors tend to grow more or less slowly, but usually once they are in a growing mode, they tend to grow over time. So that's an important point. And the major consequence of this observation is that since most of the trials that have been done so far do not include a placebo arm, meaning a control population that is not taking the drug, the idea would be to use the natural history study, at least to be a reference, a control population for some of these future clinical trials, especially those focused on hearing preservation.
Yeah, I think this is
that is my take on that.
Yeah, super important
study.
point. Yeah, super important point you're bringing up is, in fact, even in the SPRINT trial, which is the trial that has led to the approval of Koselugo, the natural history of the tumor growth was actually used as the control arm. So the importance of these studies has to be really stated, because that will allow us to actually give everybody drug in a later stage, right? That we don't have to give people placebo, especially for a rare disease that is so incredibly important. The other thing I heard during the meeting is, also the evolution of the imaging technologies now have made us understand that some imaging technologies allow us to maybe measure tumor growth, but may help us now understand that some more complicated imaging modalities to which we have access now are actually able to even understand hearing loss versus non-hearing loss, right? So with that, I would like to shift gears again. I think, Dr. Giovannini, you made an important point about the topic that was brought up during the meeting, and there was a pretty large discussion about pain in schwannomatosis. That whole idea of that there is this macrophage infiltration, so these cells that are, again, going back to your tumor heterogeneity, these macrophages that are giving you inflammatory response, and that may be some of the pain. What is that? What does that mean for how is that going to help us identify now mechanisms that are, let's hopefully, druggable for our patients in pain with schwannomatosis in your hypothesis?
That's a very important and crucial point, Annette, because I will try to make it extremely simple. We know that the molecular mechanisms that are responsible for schwannoma growth in all types of schwannomatosis, so NF2 related and non-NF2 related, depend on the loss of the NF2 gene. So from that point of view, and that's what I keep repeating, and it was definitely discussed in depth at the conference, is based on this observation, a non-NF2 schwannomatosis schwannoma could benefit from a drug or a therapeutic intervention directed to an NF2 related schwannoma. So the idea of eventually in the future opening a new enrollment to non-vestibular schwannoma in a specific basket of the Intuit trial, for example, and that, but Annette has more information on this, on what's going on in Europe, where that has been already decided. So as far as tumor control, I think we can now safely consider all schwannomas across all schwannomatosis as potentially able to respond to a drug that we identified by studying NF2 only. As far as the pain, the situation is more complicated because we still don't know exactly why there is this strong specificity of schwannomatosis or non-NF2 schwannomatosis schwannoma to have a pain phenotype associated in sometimes the tumor is painful, sometimes the nerve is painful. So, but at the same time, there are also pain manifestation in NF2. So the situation is not perfectly black and white, but definitely there is a predominance of pain manifestation in non-NF2 schwannomatosis. And that could be due, first of all, to the additional mutations that are present in non-NF2 related schwannomas, for example, SMARB1 mutation, LZTR1 mutation, that could by themselves modify the cells, the glial cell, the Schwann cells, or even the neurons to produce factors that could induce pain. And the other point that Annette suggested to is that it is now clear that there are different percentages of macrophages infiltration in schwannomas and in meningiomas. So that in combination with those additional schwannomatosis specific mutation could lead to a pain phenotype. But again, this is very new. It's interesting that a specific technology was able to find this, to discover the presence of these blood cell types like the macrophage in the schwannomas and the meningioma. And the technology is called single cell RNA sequencing, where it became clear that there was a signature that we normally see in blood cells and not in nerves, for example. So that is something very important because it's also, it's something that the research will be focusing more and more in the next few years. So you will see more proposals, research idea, wanting to understand what is the heterogeneity. Again, this is an element of heterogeneity, how many macrophages there are in a tumor versus another one. So that makes already two different tumors that are likely to respond to different treatments in different ways.
Super, super interesting. I saw that there were some questions in the chat. So I don't know if we want to jump into the questions. Maybe what I did for everybody is I put in the address for the NF2 longitudinal natural history study. It's very simple. It's nf2@nih.gov. And we will definitely also make sure that everybody who is in the NF registry from CTF will be informed of this longitudinal natural history study.
Do we want to take a minute and just recognize some of the patients who were at the meeting? And
absolutely, and maybe, and also, yep, absolutely.
And then we can get to some questions.
7:40, so I was like, okay, I'll wait until 7:40.
We still have a few more minutes, but let's
super important. I think what would be really important also is to hear from the patients their perspective of the, I'm sorry, this is my daughter, I apologize. I also would like to hear from the patients their perspective of the meeting.
So that would be, I know we have on the meeting, we have Clayton Molina, Sheila Heel, Barbara Franklin, Camille Toms, and Michelle Holbrook. It's an amazing group. We were so excited to have all of them and a few more actually at the meeting. So it's hard to determine who should go first. Why don't I ask Clayton? Clayton, I think you're on. Why don't you give a little input as to what the meeting meant to you? What were some of the great takeaways that you had from the meeting, particularly knowing that you work in the biotech space now as well?
Yeah, thanks. Yeah, it was my first time to attend the state-of-the-art meeting. And of course, it was the first time that meeting had been held in quite a while due to the pandemic. And it was very, very gratifying for me to be there and learn quite a lot about the science that is going into the search for therapies and ultimately a cure. I really enjoyed the time. I think I would just say to any patients in the meeting, you know, if it's interesting to you to get involved in that level and interact with scientists and sort of just dive in, kind of probably beyond comfort zone, at least for me, for myself, it's definitely rewarding to do so. And in terms of what excited me about the content of the conference, I really kind of echoing what some of the topics that were just discussed. But I think that overall, the level of sophistication for disease models was really exciting, and it came on multiple fronts. You know, it's necessary to have some type of model system, whether that's a mouse model of schwannomatosis, or a cell model of schwannomatosis, or some cell line that, you know, has some aspect of the disease, because those are the type of tools that really enable researchers to search for cures. And so, yeah, I think there was some really interesting work, I thought, relating to in vitro models for schwannomatosis, in particular, work to create Schwann cells, you know, in vitro in a test tube for screening drugs or doing other basic science experiments. So really interesting work there that would be able to really expand the scale that drug discovery that can be applied to schwannomatosis. And of course, we saw a really exciting example of mouse model for ependymoma, which is you know, enabling researchers to understand that manifestation of the disease. And of course, I would be remiss not to mention a great news on a pig model from Marco's lab, which is quite exciting because, of course, pigs are very like humans, so very good model system to test drugs. And I think that Marco's comments and overview in the previous comments on the tumor heterogeneity are quite connected to this, right? You know, the picture is that the tumors, they're not just one type of cell, they're actually very complicated small ecosystems. And that's exciting because as we understand that complexity, it opens up lots of other opportunities for therapy. You know, there are opportunities to consider therapies that target different cell types that are within the tumor you know, heterogeneous environment. So, you know, just really exciting to see all the progress on that front. And I came away with really positive kind of outlook based on what I heard about those things.
Yeah, I think this is thank you, Clinton, for saying that. It is, of course, extremely important that once we understand the biology and start understanding which mechanisms we can inhibit or activate, then, of course, you need some kind of selection system, some kind of filter system to say, I have a lot of drugs or potential future medicines here. I need a filter to test them to bring the very best of those drugs to the patient. So the development of all these model systems are, of course, extremely important. And we had a lot of creativity of new model systems that will be developed. And I hear you think, why pigs? Pigs are really important because exactly what Clayton said, they look very much like humans in the sense of their metabolism is the same. You've probably heard of people that have, you know, transplant pig hearts. So it's very close to the human system. So if we want to look at how a drug would respond in a human body, you could actually use a pig to predict that. So they are super important animal models to make sure always the same, right? That we bring the very best medicines to the patients. Michelle, back to you. Next,
So I
the
just want to
next
introduce.
patient who want to say something in case they want to.
Yep, no, we have a few more patients. I just want to make sure that they have a chance to talk a little bit about what the meeting meant to them, because we do have some questions in the chat that I want to get to. And I just also want to mention that Clayton also works for another NF2 patient, Faber, who's been in the NF2 space for a very, very long time. So
And who was at the meeting? Yeah.
Lucky to have so many amazing patient advocates and representatives out there. I just want to have Camille Toms be spotlighted for a moment, so as a patient who attended a part of the science meeting, but the patient meeting the day before the science meeting started, just to say a few words about what being there and what the meeting meant to her.
I'm sorry, I'm here. Hello, everybody. Yeah, I attended the scientific part of, I believe it was Friday of the meeting, and it was a lot of information. It was really good information. I'm glad that we are putting as much research as we are into this condition. Is it still called NF2 or is it called a different name now? I don't know. I'll find that out later.
It's NF2 related schwannomatosis, it's called now.
All right. I think it's really interesting that we're working on pigs as they're like humans. It was interesting to know some of the, new, I learned new information about my eyes there that I didn't know before. Because I met her there, I forget her name, the neuro-ophthalmologist that I met there. But she had really great insight into some eyeball issues I'm having. Yeah, but it's just really nice to see a whole team of people working together to cure NF2 without, and thinking about the ways to treat the tumors that aren't always necessarily surgically related, because that's very hard to go through. On my behalf, it's very hard mentally. I go through the surgeries, but then after it, it's a big mental big mental game to play. So yeah, I think something that was said there was like, you can't cure NF2 with a knife, but I'm on Avastin, but bevacizumab, I believe.
Yep.
And yeah, it's really interesting because I've been on it for 2 years, and I'd like to know if it's gonna not the best thing to stay on, or other things you could do. And the vaccine sounds promising, so very exciting.
Thank you, Camille.
You're welcome.
So the other topic I quickly wanted to throw out was there was an interesting discussion on pain in schwannomatosis, and we have been trying to develop a platform trial in Europe. Well, we're almost done with it. I hope next spring we should have a platform trial in Europe. But the platform trial for schwannomatosis pain was complicated because there was somehow a misunderstanding of what kind of pain our schwannomatosis patients were actually suffering from. So I thought Michelle Holbrook had some very interesting insights on that part. So I don't know, Michelle, if you feel like speaking up tonight. I thought it was a very insightful conversation, and really to me highlighted the importance of patient engagement, right? It is not just the patients are there because they are the clients of everything we're doing, but they're actually the co-designers of the research and the clinical trials, right? So I was very blown away by Michelle's comment during the meeting. But I don't want to throw you on the spot, Michelle.
Oh, no, sorry, I just got off a plane, so I apologize. I was in New York. you know, I think we've come a long way, but with schwannomatosis, as I stated, I have excruciating pain every day, as well as the majority of the people that do have schwannomatosis. I'm very fortunate. I was, you know, a lot of the things, because I am not a researcher, I'm not a doctor. I sometimes felt like, oh my goodness, I'm a deer in headlights, but I will tell you, the research that these doctors and the scientists and everyone involved is doing for the Children's Tumor Foundation is absolutely amazing. And I walked out of that meeting feeling positive, knowing that we will continue to press forward for NF and for CTF. And as well as going, you know, making sure that people, making sure that people register for the NF registry is very important.
For like $4.99 a day.
And we'll put the NF registry, if anybody isn't a part of the NF registry, we'll put that into the chat so you can become a part of the NF registry, because it's one of the most important things as a patient that you can do to help in scientific research and being a part of our community. And you'll get information on clinical trials that are happening. You'll get information on programs like this one tonight as well.
I
think, I think one of the things that is very important to me is trying to make sure that as a schwannomatosis patient that we figure out what we can do for the pain. And I can't thank everybody enough that was at the state of the art, all the doctors, the researchers, everyone involved, taking the time to really listen to what we had to say as a patient and taking that into account to try to press forward on everything that we're doing.
Yeah, absolutely. So I saw 2 questions in the chat.
And
So
David also has his hand up, but let's get to the questions in the chat and then David will come back to you.
okay, let's do one question and then we get to David. Does that make sense, David? Is that okay for you? So there was the imaging question, Dr. Nymfu. I'm going to throw that question at you.
Sure. Yeah, so I think there was a question about how does imaging predict hearing loss? And I would say to start that it's still a ongoing research, and that is part of the talk that was given about the NIH researcher, Dr. Prashant, who talked about the natural history study.
Exactly.
So through the natural history study, they were able to look at imaging and found that in patients who had something called flare signal changes. So flare is a type of sequence with MRIs, and these are very thin cut MRIs that go through the internal auditory canal. So really where a vestibular schwannoma would be. And if they see this abnormal signal from T2 or flare kind of more into the cochlear portion of the hearing apparatus, then that somewhat predict that patient would have loss of hearing. It is a little bit retrospective, right? Because it cannot predict when that will happen. It's a little bit more of a study that when they see that happens, then it correlates with somebody having hearing loss. And it's still a small number that they had in order to do this. So obviously, I think being part of a natural history study, or if a patient is on a clinical trial study, for instance, like with Intuit, even when they're not on treatments, we still try and keep their history, because they're actually part of something called a master study on Intuit, where we continue to keep their history, their scans, their tumor tissue, to look at any changes after they've been on a treatment, or what happened to scans over time, because we'll need more number to really prove that is a biomarker, imaging biomarker of hearing loss. So it's just one of those things. So I think one of the questions too is like, where do you get these scans? And I would say, being a clinician, I think it's always hard for patients with insurance and things like that to get scans at one of the NF centers like ourselves, where we actually develop very clean cut and high resolution scans to make sure that we look at areas where we would expect tumor in NF2. For instance, thin cuts through the intra-auditory canals, for instance, versus going to just any regular imaging center. They're just not going to quite do scans the way we think that needs to be done in order to use a scan for 3D measurements, right? I think that was another question about 2D and 3D. 3D volume better, but we can only do that if there is enough images in order to make a 3D reconstruction and volume size of a tumor. So that's really kind of I'm trying to answer actually multiple questions at once, but like I said, still early finding, and it's important to get good scans in order to do that. It's not any one specific place. I've seen scans from my other colleagues who are in NF centers, and we're all pretty similar in that we try and do higher resolution scans.
Cool. Thank you so much. David, I want to make sure that you have a chance to speak up.
Hello, everybody. My name is David Clauder. I have schwannomatosis. I was diagnosed back in 2000 by Dr. Catherine Nathanson. She was my genetics doctor at University of Penn. But I live in Myrtle Beach, South Carolina now, and I've had 2 surgeries on tumors. One on my L5S1. It was a tumor in that nerve down there. At the time when I had it back in Philadelphia, at Jefferson University, I didn't know I had schwannomatosis, so they tried to resect it. It didn't completely come out, and they had to do another resection, and they didn't cut my nerve, which was good, but there were still parts of the tumor in there. But I didn't know I had schwannomatosis at the time. Then I got hit in the arm. I had a shocking pain down my arm, and then I went to a neurologist, and they said, let's get MRIs. And then that's when the doctors noticed all the tumors. And then I went to Dr. Nathanson and got diagnosed. Since then, I actually had brain surgery back in 2016. I hadn't got an MRI since 2010. I know I had a spot in my brain, and I hadn't followed up on MRIs for 6 years or whatever. And then I started getting pain in my left eye, and it was like a needle going into my eye. I went and got an MRI, and the tumor, make a long story short, the tumor blew up. So I went to MUSC, Medical College of South Carolina, and I had brain surgery from Dr. Vandergrift, and he pretty much saved my life. He took the whole tumor out. There's no tumor left in there. And there is, my face is numb, but there's no muscle nerve cut. My question, I'm at a crossroads right now because I have a tumor in my left arm. And I don't know if it's worth me waiting around for therapies to shrink this tumor or to just go ahead and get the surgery on the tumor. That's with all this research, and I just don't know if I should wait around. The pain, it gets worse. And I know that the doctor talks about how the tumor grows like a tree, so it grows slowly. But if somebody bumps it, it's very painful. And over time, it just causes a lot of pain. So
David, I'm sorry to interrupt you, but we have to be careful that we do not address personal cases. We can get you in touch with a clinician to talk to, and I'm pretty sure that maybe the clinicians are willing to talk to you on a separate setting. But I think we have also, for privacy reasons, I think we have to be really careful that we do not address personal medical questions on these webinars that are going to be recorded, distributed, blah, 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 right? So even you want to be careful there, but we will make sure that we get you in touch on a private call, but we prefer to be careful with personal medical questions.
Okay. Hopefully I gave some kind of insight on what.
Very interesting, but we will make sure that somebody reaches out to you. We just don't want to make this a public, we don't want to make your case a public situation, right? You don't want to respect your privacy.
There are 2 more quick questions, and I know we just have about 3 minutes left before we need to close out the evening.
Yeah.
We want to give Barbara a minute or 2. Do we want to address one more question in the chat, and then we'll ask Barbara to say a few words? I mean, the two questions that were left, one is there any work being done on the complementary pathways to VEGF like Notch or HIF1 inhibition? And then Tony also asked, has anyone seen tumor shrinkage without radiation or pharmaceutical treatment?
I addressed the Notch HIF1 question in the chat, so briefly.
Yeah,
Great.
we are keeping, I'm just going to check 1 second, but I am not sure. We can take this offline. I know I don't think Notch have been addressed. I worked on Notch on my previous job, and there were some challenges with toxicity that I remember, but I'm more than happy to look a little deeper into that, because we are, just for everybody on the call again, we are keeping a list at the foundation of all the new molecular mechanisms that have been published in NF1, NF2 and schwannomatosis, or should I say NF2 related schwannomatosis and the other schwannomatosis. So we will check that, and we will get back to you as soon as possible.
I think on the question of has anyone seen tumor shrinkage without any radiation or pharmaceutical, and I also assume surgical treatments. And it's actually a little bit unlike NF1, where we actually, in those cases, sometimes see some of the tumors actually shrink spontaneously as a child grows up. We actually don't see that as much in NF2 related schwannomatosis or other schwannomatosis. So they may stop growing, they may remain very small in size, but we don't see them shrink without an intervention.
So I just looked it up for the person who asked the question about the HIF1 and Notch. There is not that I see, as far as we are looking through the PubMed literature, there is nothing for NF2 out there. There is HIF1 literature around MPNST, but I don't see something clear that pops out that came out of the PubMed searches so far for NF2. Barbara, I think we should not be right if we wouldn't give Barb Franklin.
Of course, and I did tell her we would have her say a few words at the end. So Barbara, do you
and
you and Sheila
and our former chair Tracy Galloway, right? I think it should Tracy should close. Barb, go for it.
Yeah, I mean, so much was covered, so I'm not going to repeat it, but there are a couple interesting things. One that was touched upon lightly is looking at patients that have been on Avastin for a long time, and the surgical outcomes for some of those patients was surprising to surgeons because of the blood. And it really stressed the need for a surgeon who has the experience. And they are now embolizing the vessels before surgery even begins to avoid that. So it really does become important to see an NF2 surgeon. Marco and Leah, I have to say thank you. This was so overdue and necessary because, you know, no matter what we've all been able to do, including doctors and researchers, it never equates to the face-to-face. And seeing the breakouts and people talking and the continuation of important topics really was valuable. And I thank all of you for that. The ependymoma discussions I thought was very interesting because, you know, as I always refer to myself as the grandmother here, the longer patients live, these what used to be designated, you know, innocuous ependymomas, nothing's going to happen. It's because patients died. They could, and now with patients living longer, these tumors are growing, and even if they're not growing, the compression is enough that it is causing problems. So there was interesting debate. I can't say there was a decision, but there was interesting debate, and again, very important to have a doctor that specializes in these spinal tumors. Because they're definitely, and there is mixed consensus you have to be comfortable with. These doctors, some were saying that they actually, before symptoms really even evolve, if they see any growth, even without a symptom change, they want to take it out because once the symptoms show up, you can't get that back again. Yep. So, you know, that was pretty strongly debated, but made some sense. And they talked about the cysts on ependymomas, and they are different. and they don't get treated successfully with any drug, maybe Avastin, who knows, but they present their own pressure. So I thought that was important. Same thing with radiation. I was shocked to hear the mix and kind of negative on proton. I always had in the back of my mind that was going to be the safe one to use. And there was a real mixed consensus on proton or any kind of radiation. And I guess it was just interesting hearing some of them talking about some of the pathways like HIPAA. And Sheila and I would both say the VEGF vaccine is what got us so. But, I also must say, thank you, UCLA. It was an incredible event. And Marco, seeing this group of specialists, I mean, the woman who talked about facial nerve and her expertise was incredible. I've always had one woman in mind, and now to know not only is there competition, but great expertise from her. And just the whole group that you've got there, it's great to hear the West Coast really has a strong center now. But those were my top lines, nothing else I think.
Thanks, Barb.
Thank you, Barbara.
And Annette, I just want to add a quick information on the gene therapy approach, because I'm getting a lot of questions on that. And I remember discussing with Barbara extensively, after we heard those talks. And of course, there is a lot of interest, both on the patient and in the scientific community about the potential application of gene therapy to NF2, NF1, schwannomatosis, to all of them. I think that we have seen that there are several teams actively engaging in research on, giving a proof of concept whether reintroducing the NF2 gene, for example, would be beneficial for these patients and to control tumor growth or to stop or shrink tumors. So there is some interesting evidence, but of course, it is such a challenging endeavor to reintroduce a gene in all tumor types and potentially in all tissues from where schwannomas, meningiomas, and ependymoma originate in an NF2 patient, that we are really at the prehistory of that approach. So again, there is a very strong commitment in the scientific community and in the clinical community to explore this opportunity, but I just want to give a realistic, expectation on what we can see within the next 5 to 10 years.
Yeah, super important.
Much more investment, much more information, and hopefully the technology will improve as we move forward. Because I like to make the analogy on the how fast they were able to develop a vaccine for COVID in the matter of 2-3 months, actually. And that was just by combining different technology that had evolved independently, and suddenly by mixing all together, they were able to come out with this very important tool that basically ended the epidemic. So I think we need something like that. We need research in different areas, in the area of virus, vectorology, on protein expression system, and to make it real. But for sure, it's an option. And I would say that so far at the horizon, the potential of gene therapy and or immunotherapy for NF in general is probably the most exciting, but also high risk and high challenge options that we will be seeing in the next few years.
No, I thank you for making that comment, Dr. Giovannini. I think the COVID has indeed really taught us that a portfolio approach, whether it's a small molecule or an antibody or a gene therapy or all RNA or whatever else, that in fact it's the portfolio approach that will really help us for moving forward. And I think that has been the approach of CTF, and we're not planning to change that, honestly. I know that, and I want to be respectful of everybody's time. Michelle, I know you wanted to make a couple of closing remarks, so I'm going to move the microphone over to you.
Thank you. Well, first and foremost, I just want to thank everybody who attended tonight, and thank you for all the amazing questions and conversation in the chat. This has been recorded, so we will be sending out a recording and the transcript in the next few days. Just give us a day or two to review it. So we'll make sure that you have that in your inbox. I also want to do a shout out to Emily Crabtree and Amy Boulis, who are behind the scenes tonight. They're staff at CTF on the development team. We also have Salva LaRosa, who is our chief science officer, on the call tonight, and Simon Vukel, who is our chief marketing officer. Thank you all for joining us. And then, of course, a huge thank you to Annette, to Dr. Niemfu, to Marco Giovannini, and to our patient representatives, Clayton Molina, Barbara Franklin, Camille, Michelle Holbrook, and I'm sorry, Sheila, that we didn't get you on the camera as well, but we will do that in the future. So thank you everybody for joining us. And if you want to become an NF2 accelerator, which is being a patient advocate for us and telling your story and helping us spread the word about what we're doing in NF2, if you're not already an NF2 accelerator, just email nf2@ctf.org and we'll make sure that we get you to be an NF2 accelerator for us. It's a lot of fun. Most of you on the call tonight are, and we want to build and grow our NF2 community so thank you for helping us do that. And I believe that's it. And with no further ado, can say good night.
Thank you, everyone.
Thank you so much. Thank you.